Okay, this week we'll be talking about the basics of genetics, which is an introduction to hereditary traits, and we'll be talking about genetics and heredity for the next couple of weeks. So please make sure that if you don't understand these concepts at the end of all the activities this week, you spend a little bit more time reviewing, because if you don't understand this week's concepts, you'll have a much more difficult time in the next couple of weeks. Okay, heredity is the way that organisms pass traits or characteristics on to their offspring. These traits, as we mentioned last week, are determined by the organism's DNA sequence. Although all of your traits are determined by your DNA, in biology we often track traits or characteristics that show variety in a population. So, for example, even though the fact that you have 2 arms and 2 legs and a nose is all determined by your DNA, usually when we're talking about heredity or hereditary traits and genetics, we're looking at characteristics that are not the same throughout the whole population, something that shows variety. So for humans, some examples of these traits are eye color, hair color, height, body build, blood types, intelligence, and gender. Those are some things that are commonly tracked and things that we talk about and use as examples in a traditional biology course. There are obviously all kinds of other genetic traits that biologists look at and follow. They talk about genetic diseases and autism and anything that happens to have a gene associated with it, a scientist can track. The biological field of genetics is the study of these hereditary patterns. So heredity is the passing down of traits. Genetics is the field of biology that follows those or tracks those traits. Okay, the field of genetics absolutely begins with Gregor Mendel. And Mendel was an Austrian monk who was born in 1822. He happened to have a fascination with hereditary traits, and he was an extremely detailed man and also a teacher. So he was fascinated by this field, and there wasn't a whole lot that was known about heredity at that time. So he did some studies that were fundamental in finding out the foundation of genetics that we still use today. So Mendel began systematically breeding pea plants to investigate the passing down of traits. Pea plants were something that had a lot of characteristics. He looked at many different characteristics. We'll talk about those in a second, but he could actually control the pea plants very carefully. He could take a pea plant that had certain characteristics and he could cross-pollinate that. In other words, take the pollen from one plant and put it on another plant and make sure that those 2 plants were mating so that he could follow those characteristics and those hereditary traits very carefully. They weren't going to up and move and mate by themselves. This was something that he had complete control over, and that made it easy for him to study. He collected very detailed data on flower color, pea pod shape, height of the plants, shape of the seeds, and more. So here's a chart of all of the things that he looked at, and you can see characteristics of seeds, flowers, pods, stems, and all of those things he tracked in very detailed ways, in very controlled experiments, so that he could carefully follow what was happening to those traits from one generation to the next. Okay, Mendel kept track of the traits in each generation of plants and created some terminology to do that, which we still use today. The first term we'll talk about is the first filial or F1 generation. And this is the first generation of offspring created when 2 plants were cross-pollinated. So he takes, the pollen from one, puts it on the other plant. That's cross-pollination, and this is the first generation of offspring that's created from those 2 plants. The second filial generation is the next generation. So when two plants of that first generation were cross-pollinated, they created an F2 or second generation. So here's a picture of that. You can picture these two pea plants, one pollinating the other, and then him following those traits into the next generation, which he's calling the F1 generation. Two plants from that generation create more plants in the F2 generation. And obviously there are lots of plants in each of these generations, but he usually looks at the passing of traits from two specific plants so that he can see how that information is passed down. Okay, Mendel noticed some trends that were occurring as he was tracking these traits. He noticed that when two plants with different traits were crossed, the F1 generation only showed one of the traits, and the other seemed to disappear. When two of these F1 plants were crossed, the F2 generation showed the trait from the F1 generation in 3/4 of the F2 plants, and the trait that had disappeared showed again in 1/4 of the F2 plants. So that's a lot of words, but this picture will give you a good idea of what I'm talking about here. So if he took two plants with different traits, we've got a green pea plant and a pea plant with yellow peas, yellow pea pods. When those two plants with different traits were crossed, the next generation all only showed one of those traits, in this case, green pea pods. So one of those traits, the yellow pea pods, seemed to completely disappear in this generation, and the other trait took over 100% of that generation. If he took two plants from that generation and crossed them again, then this yellow pea pod would reappear in 1/4 of these second generation plants. So 1/4 of the plants would show that disappearing trait, and 3/4 would show the trait that was in 100% of that F1 generation. So it disappeared and then it reappeared. And he saw that over and over again. He named these characteristics, which we now call traits, the dominant trait. And the dominant trait was the characteristic that could be seen in the F1 generation that seemed to dominate the other trait or make the other trait disappear. And the recessive trait is the characteristic that disappeared in the F1 generation but reappeared in the F2. So if we look at our picture again, here the dominant trait is the green pea pods, and that is overshadowing or dominating the recessive yellow trait. Recessive trait is the one that disappears and then reappears. He couldn't quite figure out why that was, though, yet. We'll get into that in a little bit. So let's spend a minute talking about genes. Mendel was talking about traits. We now know that each of the traits that Mendel studied was determined by one gene. And a gene is simply a segment of DNA. Remember, we talked about the DNA starting to create a protein at a certain point and then ending at a stop codon on the mRNA, and that segment that is coded before the stop codon is a gene. So it's a segment of DNA that codes for a specific characteristic. Considering that DNA hadn't even been discovered in Mendel's time, he created some pretty revolutionary rules based on his pea plant observations. So we'll get to those in 1 second. Let's take a look at a picture of a chromosome here. You can see the X-shaped chromosome. Each of these bands, a light or a dark band here, is a gene, and this is how geneticists symbolize genes on a chromosome. So Mendel used his information, and even though he didn't know anything about DNA yet or genes, he came up with 4 principles. First was the principle of unit characteristics. which means that an organism's characteristics are caused by units that he called factors. We now call those factors that determine characteristics genes, as I just said. Second, the principle of dominance. In cases when two or more forms of a trait exist, some forms may be dominant and some may be recessive. We just talked about that as well. Next, the principle of segregation. Segregation, remember, means separating. In sexually reproducing organisms, each adult has 2 copies of each gene. We talked about that in the last chapter. These genes are segregated from each other when gametes are formed. So way back when we talked about meiosis, you can remember that each adult has 2 copies of a gene. Those were our homologous chromosomes. Those genes are segregated during meiosis and separated from each other so that each new gamete or sex cell has 1 copy of that gene. And the principle of independent assortment, which is that the alleles, which are those different forms of a gene, we'll talk more about alleles in a minute, segregate independently from each other. That means that when egg cells are created or sperm cells are created, the genes are separating from each other. They're not separating on an individual chromosome, but each chromosome can go its own way and it's randomly allotted into a different gamete. All right, let's talk about some genetic terminology, and this is the information that really is going to be pertinent in the next couple of weeks. So first, let's say the word phenotype is our first term. A phenotype is a physical expression of a gene, for example, tall pea plants. So a good way to remember this is phenotype starts with that PH sound, physical expression or physical trait. So this is what you can actually see. A genotype is the genetic makeup of a gene, usually represented by letters. So for a tall phenotype, we might give it the genotype of capital T, little t, and we'll talk much more about that in a minute. But a genotype is a combination of letters. It's just a representation of the organism's actual genes, which you can't see outwardly. Capital letters represent dominant alleles, and lowercase letters represent recessive alleles. So an allele, remember, is a form of a gene. So in our tall, if our gene is height of a pea plant, you can have a dominant allele, which makes it tall, or a recessive allele, which makes it short. And the dominant allele is represented by a capital letter. The recessive allele is represented by a lowercase letter. If both alleles for a certain trait are dominant, for example, capital T, we call that homozygous dominant. Remember, we talked about mixtures and we said in a homogeneous mixture, it looks the same because homo means same. So homozygous dominant means 2 same alleles, both dominant. If you think about it logically, it should help you remember what these terms mean. Heterozygous, hetero, remember, means different. Both alleles for a certain trait are different. So you have 1 capital and 1 lowercase, or 1 dominant allele and 1 lowercase allele. The dominant allele is always written first. The capital letter always goes first to show that it's going to overshadow the other one. And then our last term here is homozygous recessive. Homo, remember, means same. Recessive, lowercase, so we're going to have 2 lowercase letters or 2 recessive alleles. Okay, the letter chosen for the trait is based on the dominant characteristic. So if, for example, round peas are the dominant trait that's overshadowing wrinkled seeds or peas, then we're going to use the capital R for round. And we don't use R for round and W for wrinkled. We use only the letter for the dominant trait. And then we use just capitals and lower cases. Because the dominant trait overshadows the recessive trait, both the homozygous dominant, which is two capital R's, and heterozygous, which is capital R lowercase r, will show the dominant trait. So for example, round seeds are dominant over wrinkled seeds. So capital R dominant over lowercase r. Therefore, capital R individuals will be round. Sorry, this is a typo. It should be round. This is homozygous dominant. Those will be round because they both have they both have dominant alleles or capital letters. And this heterozygous genotype, capital R, lowercase r, will also be round because it has one capital letter, one dominant allele, and that will overshadow the recessive allele. The only plants that will have wrinkled seeds are lowercase r, or homozygous recessive, because there's no dominant allele in there to overshadow those. The recessive trait can show through. Okay, let's talk about Punnett squares. Punnett squares are how we represent the combination of traits in the creation of offspring. They're a quick way to show how alleles will be combined in the next generation. One parent's alleles are written at the top of a 4-square box. So here we have one parent. This is going to go here. This is one parent for one trait. And the other parent's alleles are going to go on the side, and you separate them. So we have a capital A, lowercase a. This is a heterozygous parent. We're going to separate those alleles and put them at the top. This is another heterozygous parent, and we're going to separate those and put them on the side. And then just like in math, when you are graphing and you're bringing one part over and one part down, that's how these alleles are going to combine. And then these 4 boxes will show your possible combinations of offspring. So for this example, we'd be bringing down a capital A and over a capital A, and this offspring would be capital A, capital A. This one's going to have a capital A and a lowercase a, and that's going to be heterozygous. Remember, you write the capital letter first. This one's going to be capital A, lowercase a. Excuse me, and this one will be lowercase a, lowercase a. And this is the offspring that's going to show the recessive allele because it doesn't have any of those capital A's in it. It doesn't have any of the dominant traits overshadowing it. So let's do an example here. A pea plant that is homozygous recessive for yellow peas, lowercase y, is crossed with a pea plant that is heterozygous for yellow peas. Capital Y, lowercase y. Yellow is the dominant trait because we're using Y's. So our genotypes, here's our Punnett square. The genotypes of the offspring are going to be capital Y, lowercase y, which will give you a dominant yellow pea. And this one's going to be, again, heterozygous, which shows the dominant trait. And these 2 offspring are all going to have recessive alleles. They're going to be homozygous recessive, so they will show the recessive trait of green. So here is one parent, here's the other parent. We said homozygous recessive, heterozygous, and these are your possible offspring combinations. Now when we talk about the offspring, we mentioned genotypes and phenotypes. Again, genotypes says the offspring will be 50% heterozygous, so this is your genotype, the letter combination, and 50% homozygous recessive. So 2 out of the 4 boxes are going to be capital Y, lowercase y. 2 out of 4 of the boxes, or 50%, are going to be lowercase y, lowercase y. And the phenotypes, which is how they look, their physical expression, is going to be 50% yellow, 50% green. Okay, and I just added the pictures in here for simplicity. We don't put pictures in a Punnett square. Usually this is just a way to represent it, but we usually just use the letters. But you should know that because yellow is the dominant trait here, anything that's heterozygous is going to show that yellow trait. Anything that's homozygous recessive is going to show the recessive trait of green. Okay, so that is just a classic example of a Punnett square. We're going to do a lot more practice with Punnett squares this week. Now, as we're talking about percentages, we need to get into some probability. The field of genetics deals with percentages and probabilities. Traits are passed to offspring randomly during meiosis, so it's impossible to predict without looking at a DNA sequence what each offspring's characteristics will be. So if we go back to our Punnett square here, this is not to say that the first offspring is definitely going to be yellow, heterozygous, and then this is the second, and this is the third, and this is the fourth. That is not the way that it happens. These are the combinations that can arise in the offspring, and then we give percentages of probability. So we say about 50% of those offspring will be green and 50% approximately will be yellow. Just like in humans, the probability of having a baby that is a boy is 50%. But just because you have 2 babies doesn't mean that they will be automatically 1 boy and 1 girl, because probability is a guess, a predicted guess. It's not an automatic, perfect answer. The larger the population of offspring, the closer the percentage will be to the predicted percent. So if you have a baby, the probability that it's going to be a boy is 50%. The probability it's going to be a girl is 50%. But that doesn't mean that just because you have 2 babies, you're automatically going to have a boy and a girl. Same thing occurs when you flip a coin. The probability of getting heads is going to be 50%. Probability of getting tails is 50%, but that doesn't necessarily mean that just when you flip the coin 2 times, you're going to get 1 heads and 1 tails. The larger the population, or the more times you do it, the more numbers you're dealing with, the bigger the numbers, the closer the percentage will be to the predicted percentage. So the more times you flip a coin, the closer you'll get to 50% heads, 50% tails. The more babies you have, the closer you'll get to 50% boys, 50% girls. Or if we're talking about the whole human population, we're a lot closer to the 50% boys, 50% girls than you would be necessarily in one family. So we're talking about percentages and probability, not individual offspring, and knowing specifically what one individual's genes are going to be. We're talking about a whole generation and how many of those individuals are going to have certain characteristics. So to summarize here, because remember I said we're going to get more complicated next week, let's just summarize what we talked about. The type of heredity that we learned about this week is called complete dominance, or Mendelian inheritance, because it's associated with Mendel and what he discovered. Complete dominance is when a dominant allele and a recessive allele are combined in a heterozygous genotype, so capital letter and a lowercase letter. The phenotype will show the dominant trait because the dominant trait overshadows the recessive trait. So here's a good way to think about it. Here's our heterozygous genotype. The capital letter, the dominant trait, is going to overshadow that recessive trait. And that is called complete dominance. All right, let's get into slightly more complicated material for this week. Test crosses. Let's say, for example, you have a black mouse, which is hypothetically a dominant trait. I don't know that it necessarily is, but we're going to pretend that it is. You really love this black mouse, and you'd like to have more black mouse babies. You don't want any brown mice, you don't want any white mice, you just want black mice. Unfortunately, you can't tell just by looking at the mouse whether it has a genotype of capital B, homozygous dominant, or capital B, lowercase b. You can't just look at that black fur and say, oh, that's homozygous dominant, or oh, that's heterozygous. To determine this, scientists do a test cross. In a test cross, an organism with an unknown genotype, because we don't know if it's homozygous dominant or heterozygous, we just know it's showing the dominant trait, is crossed or mated with a homozygous recessive organism, little b in this case. If the unknown organism is heterozygous, capital B, little b, some recessive colored mice will be born. Remember, it should be 25% of those offspring that show the recessive trait, according to what Mendel figured out. If none of them show the recessive trait, then you have a homozygous dominant mouse, because just like those green pea plants, if you have a homozygous dominant mouse, all those dominant alleles will cover up the recessive ones in the mouse that it's mating with, and then none of those offspring will show the recessive trait. So based on the offspring, you should be able to figure out what is the genotype of that mouse that you have. And that's how a test cross works. Obviously, there are times when a test cross is unethical. We are not going to test cross humans and mate them with a recessive trait person to see what the offspring look like. So in this instance, you might be able to determine whether an organism is heterozygous or homozygous dominant by looking back through previous generations. So you need to go back a couple of generations, not just one, remember, because that recessive trait can be masked by the dominant trait. But if you're looking back through several generations, then you can determine some things about an organism's genotype, or at least make some logical guesses. If no recessive phenotypes exist for several generations, then it's likely that that organism is homozygous dominant, because if there were any recessive alleles in the previous generations, they would show up at some point, and you'd be able to see a recessive trait. If you don't see any recessive traits for several generations back, it's probably not there. This can be used to determine pure breeds of animals, and this is just one of the ways that we use genetics in our everyday life. We'll learn about a lot more in the upcoming weeks. But purebred animals are ones that have certain traits that can be counted on generation after generation, because there are no recessive alleles for the whatever the unwanted traits are for that animal. We use it for dog breeds, we use it for horses, and sometimes even plants. Okay, so this is your summary of genetics for this week. Make sure that you do all the practice problems this week, because remember I said it's important to understand what we're learning this week for the future weeks that are coming up as they get more complicated. And we will stop there.